பண்ணிட்டேன் மேம்.
ஓகே,
இன்னும் லோட் ஆகிட்டு.
All of you, please mute your mic. When you are answering, you can just open your mic. That's all. Shall I start?
Yes, ma'am.
Okay. Very good afternoon, friends. As you people are nearing your exams, it is time for you to start your revision. So just to support you in revising all those earlier studied material, we have arranged for these revision classes. So all these revision classes are based on your recommended prescribed textbooks. So most of the slides will be having exactly the replica of your textbook so that you can recollect it easily. So just make a rush of this revision class. First one, we start with DNA. So we do have genetic material either as DNA or RNA, and very few with protein as genetic material. You know the example for all these things. So directly getting into the DNA molecule, just we do have 4 different bases, and nucleosides and nucleotides are the basic units of it. So you know the difference between nucleoside and nucleotide, actually where it is getting differed. Okay. Apart from all these things, like these bases are arranged in pairs just by holding together using hydrogen bonds, and you should remember the bond width of all these bases also. Okay, so all these bases are held in stacks and arranged as a ladder in a double helical structure, and these bases are having a little bit rotation kind of arrangement, and they will be following a central fixed axis. And just by their helical nature, they develop minor groove as well as major groove. So you can see the difference between the minor groove as well as major groove. And these 2 different grooves are having its own capacity to have the donors and acceptors, and having different kind of exposed van der Waals forces outside the helical structure. And just because of its nature, it is facilitating the binding of other protein molecules, means the DNA binding proteins, to interact with these molecules so that the DNA is getting expressed. And you should remember the lambda of this DNA, means a minor groove plus a major groove is known as lambda of the DNA, and it is the turn. Actually, one turn takes almost 10 bases, 10.5 bases we say, and more than 11 in case of ADNA, and it differs with the model. Okay, so you just remember all these facts. And we do have 3 different models of these DNA structure. One is the bDNA, that is very common, and aDNA is the dehydrated form, whereas the zDNA is the stretched form of the same molecule. So, and you know that there are different sets of codons which is directly coding for the amino acids, and almost this is universal, and it has very minor changes, and the changes are very unique. And you do have 3 stop codons as well as 1 start, that is methionine starts. And what actually happens is the first 2 bases actually takes place, means first 2 bases takes a major role in deciding its amino acid being coded for that, whereas the 3rd base is having less role. And sometimes we can see that the purine and pyrimidine located at the 3rd base is having a different kind of coding. Okay, so all these things you can remember easily, and when you study about the transcription translation, it will be more clear to you, just because where it goes inside the ribosome molecules, and it takes its structure. The first two bases fits in, means the third one will automatically fall into the place. And all these strands are exactly packed in a chromosome. So like DNA is packed, highly packed in a chromosome. Actually, in a chromosome means one double helix is placed. It is not like so many helix are just dumped inside the chromosome. It is just only one double helical structure, and this is highly coiled using the histone proteins. And these histone proteins forms a tetramer as well as a tetramer and a dimer. And these tetramer and dimer forms actually a nucleosome core. And this core and linker DNA is formed by just by coiling over them. And this is highly packed in different models, means some different models explain this, how it is packed. And you have studied all these things in detail. So again, coming back to these 4 bases, so you know that there are 4 bases present in DNA. And all these bases are having the specific molecular weight. This we have already discussed in earlier discussions. And you should remember these kind of values for almost all bases. Okay, so just keep that in mind. When we see the double helical structure, it is all phosphates, means nucleoside phosphates are present, like monophosphates are present. So how these phosphates are formed? You do have a phosphoric acid molecule as well as the deoxyribose and the base. And when they react, there are 2 water molecules getting removed from this, and they form a deoxy nucleotide or whatever it is, monophosphate is formed. Okay. So now it is a nucleotide monophosphate is formed. And now you are seeing the elaborate a single strand of the structure. Okay. So now you can get all those things in your mind, like keep all these things in mind. So for a single strand, there will be a complementary strand in the DNA, and this strand is being means continuously growing, means it is all being added in its replication phase. And while adding, there is one deoxyribose triphosphate is being added, and polymerase is in action at last, like a pyrophosphate is removed from this reaction. And how it is taking place is it is based on the incoming molecule, whether it is a diphosphate. If it is a diphosphate, a monophosphate is added here, and the one single phosphate molecule is going out. Whereas if it is a triphosphate molecules, there will be a pyrophosphate coming out, and this pyrophosphate splits into 2 phosphate molecules going out. So it all depends on the incoming molecule and its energy level. So this is the very famous photo 51 of Rosalind Franklin. Actually, Rosalind Franklin and Ray Gosling has published this in 1953, but it was taken in 1952 to 53, I think. Later, this image was, not later, I think simultaneously, it was used by her boss Wilkins and other people, means during her absence in her lab, during her sabbatical, it has been taken out. And what you see here is, so all those stories you might be remembering. So if it is not known to you now, it is not the time to read now, because you can read it later. That is not in that much important for your exam point of view. So the darker region, what you see is the purine and pyrimidine bases tracked in the learner, and whereas the outer arc is actually the sugar phosphate backbone, and turns what you see inside is just because of its rotation, and almost 10 turns you can see in time period of 3.4 nanometer or something like that. So if you want to understand this very clearly, you can read Lucas paper, okay, 2008 Alamander Lucas paper. You can read more about it. Okay, the central part is the actual axis of it, and it shows exactly transaction view. I mean, so it's a TS view of that helix. So again, you can recollect the structure like what you have seen in the crystallography and this one. And if you see that paper, what I said, like Lucas paper, and you can compare this B and aDNA in that. We do have major and minor groove in DNA, and there is a difference in right hand and left hand helix. These two are same kind of turns, whereas the Z is differing in their turn. So this you should remember. And this page you should read carefully. So just go through it once. Just like that, this table also, some of the information given in this table, you should keep it by heart. And one thing you should note is rotation per base is given for single base in A and P, whereas it is given for the two bases for zDNA. So take note of it. And when coming to the minor and major groove, actually it is exposing hydrogen donors as well as acceptors, as well as some of the specific groups which is getting reacted over the other molecule. So that makes the difference. Like the same molecule which is present in the minor groove will act in a different way in the major groove. And just by these properties of this minor groove and major groove, proteins are getting reacted with a double helix. Can anyone tell me about the classical experiments of these nucleic acids or the base sequence, which confirmed that the nucleic acids are the genetic material? I need someone to narrate it.
Ma'am?
Yes, please, do it.
Ma'am, I will explain about 4 experiments based on genetic material, ma'am.
Yes, please, do it. Go ahead.
Can I explain with PPT, ma'am?
No need.
Okay, ma'am.
You just go through your PPT and tell us.
Yes, ma'am. 1 minute. Ma'am, நீங்க உங்க PPT ஆஃப் பண்ணீங்கன்னா விசிபிள் ஆகும் மேம் என்னோடது.
Okay. Do it fast.
Yes, ma'am. Ma'am, is it visible, ma'am?
Yeah, it's visible. And make it in this one, presentation mode. Yeah, go ahead.
Yes, ma'am. Firstly, I will explain about the Griffith experiment. In this experiment, Griffith discovered the phenomenon of transformation through his studies on Streptococcus pneumoniae. Earlier, it is known as Diplococcus pneumoniae, which causes the pneumonia in most of the mammals. There are 2 strains of virus are present, one forming the smooth colony, the other forming the rough colony. The cells of the smooth colonies are virulent and the cells of the rough colonies are avirulent. When the live cells of the avirulent strain injected into the mice and they didn't suffer from pneumonia and they are survived. When live cells of virulent strains injected into the mice, all the mice died due to pneumonia. When the heat killed virulent strain injected into the mice, they are not affected and survived. When the heat killed virulent strain with the avirulent strain injected into the mice, they are died due to pneumonia. Because some of the cells of avirulent strain changed into the virulent strain due to transformation of some of the genetic components. Next, I will explain about the experiments of Hersey and Chase. This experiment led to the universal acceptance of DNA as a genetic material. Hersey and Chase labeled T2 DNA and protein in 2 separate experiments. Therefore, labeled T2 DNA with phosphorus 32, while the proteins are labeled with sulfur 35. There are 2 labeled T2 phages are allowed to infect separately for 10 minutes. The progeny phage particles obtained after lysis. These E. coli cells contain only phosphorus 32 in phage DNA and sulfur 35 present in progeny particle is negligible, almost nil. Those studies of this experiment showed that DNA is the genetic material. experiment. Next, I will explain about the Frankel, Conrad, and Singer. This experiment shows that RNA also as a genetic material in TMV. This experiment done in two forms. In first experimental form, RNA of TMV and proteins separate chemically, and they are allowed to infect the tobacco leaves separately. Only RNA fraction of the TMV is producing the disease, not protein fraction. In another experiment, strain of A and B of TMV produce distinct symptoms on a variety. Those construct two types of hybrid virus. There are RNA from strain A and protein from strain B. In another form, RNA from strain B and protein from strain A. Those two hybrid particles allowed to infect tobacco leaves. Finally, the first type produced the disease symptom of strain A, and the second type produced the disease symptom of strain B. Hence, this experiment proved the DNA of TMV has um, DNA of TMV has have a capacity to produce a disease and as a genetic material also. And next, I will explain about the measles and Starr experiment. This experiment supported that Watson and Crick's hypothesis that DNA replication was semi-conservative. Nitrogen is a major constituent of DNA. In this experiment, E. coli was grown for several generations in ammonium chloride with N15 isotope medium. When DNA is extracted from their cells and centrifuged on a salt medium like cesium chloride density gradient, and the DNA separates out. Nitrogen 15 isotope medium has a higher density than nitrogen 14 medium. These two medium was mixed by them and allowed to divide. As DNA was extracted periodically after 40 minutes. Extracted DNA is one strand having the nitrogen 15 isotope DNA and the other strand having the nitrogen 14 isotope DNA. So this experiment shows that DNA is a genetic material. This picture clearly explains about the DNA is a semi-conservative replication. Ma'am, I finished, ma'am.
Okay. So now you quit from your presentation so that I will do it. I'll continue that.
Okay, ma'am.
So one of your friend has explained we are continuing now. So the classical examples already you have seen, like the major one, what she has skipped is to mention about the Lytic and lysogenic cycle. Okay, so the difference between these two is one is forming episome, whereas the other one is in the virus shedding phase. Okay, so just try to understand this cycle and just go through that. And semi-conservative model also she has explained, and this also she has already explained. So we are quitting all these things. Then we are moving to RNA. RNA is a little bit different from DNA, and mostly it is present in single strand. Though it is a single strand, actually it is making a random binding with its own strand, making it double helical or making it even a triple strand kind of structure. And the major difference is in one of its base, T is replaced with uracil, and apart from that, there are so many unusual bases present in RNA. And just like that, this famous structure you might be knowing, and you just remember that anticodon end is present in this RNA molecule. Okay, so you should know that where is the codon is present. Okay, so this will be making it very clear. So just remember this picture with anticodon end, and its acceptor arm is with a 3', whereas this is epsilon, and whereas this is the D loop. So normally a single strand RNA, when it is actually making it's a secondary or tertiary structure kind of thing, it forms stems and loops kind of structure. And this stems and loops differentiates from a functional as well as non-coding RNA. So this is the major feature of non-coding RNA, where the stem loop structure as well as the bulbs of unpaired regions forms a specific binding kind of properties with other reaction complexes. So this we can see when we are talking about a non-coding RNA. And just like that, it is having, I said like it is having unusual bases, and just like that, unusual base pairing is also common in RNA molecule. And this is a linear structure where we do have the difference in DNA to RNA is in its sugar molecule. Okay, so this you should keep in mind. And just like that, we are moving to the central dogma, and central dogma starts with the DNA, and then after that it is a transcription, and then at last it is producing a functional protein. And this keeps on adding the amino acids, and at last, like you can see, like a growing polypeptide chain from this, and this is a polysome structure. And just like this, we do have one more technique, polysome profiling, where the intermittent regions are chopped and polysomes are captured to have the actually regions of the mRNA. Okay, so I asked so many times about the hybrid molecule of DNA and RNA, where it is present, and here it is present. This is present inside the RNA polymerase, and this structure you should remember, like this is the enzymatic synthesis of RNA from the DNA template. And this you can go through that. RNA is actually synthesized inside the nucleus, whereas it moves to the cytoplasm over a period of time. And this is a time-based reaction which is being conducted in tetrahymena. And the summary, please kindly go through all the summary pages. And after that, some phases you should remember. This is Korenberg, and then Okazaki. Okazaki means it is Okazaki fragments you should capture in your mind. And this is Michelson style experiment. And Barbara McClintock for the jumping genes. And at last, we are moving to the base tautomers. And the difference between the base tautomer means actually the normal one and its tautomeric version is in C, it is from amino group to imino group. This is the difference. Actually, the single bond is getting converted into a double bond. And whereas in guanosine molecule, it is a keto group is converted into enol group. So keep all these things in your mind. And there can be some questions based on base flipping. In a normal double helical structure, there can be a chance of getting a base flipped out of its double helix, and that happens by damage in its hydrogen bond. And here you can see in this image, you can see a base being flipped out of its double helical structure. And the propeller twist is this turn they are talking about. What they mean by a propeller twist is this kind of turn. Normally it is parallel, whereas when it is twisted, you can see this kind of twist. And these two pages, I request you to read it again, because most of the time there will be questions based on these images. And there are coiling and supercoiling and the direction of its removing. So all these things, topoisomerase action, all these things you should remember. And when you're running a supercoiled molecule along with a simply coiled molecule, it will be getting separated in electrophoretic gel. Just by that, just by running a intact DNA molecule also, you will be getting bands on your normal gel. And while running your gel with ethidium bromide, the intercalation happens, and this is the intercalated ethidium bromide molecule. So you can easily remember where the ethidium bromide binds. So just go through this table. This is much more important. And here we do have the splicing and other, it's a splice variants and alternate splice form and coding and non-coding variations. All those things happens just by this splicing. Splicing actually contributes up to even speciation, and this we can discuss later if you need more information on this, but may not be of your exam point of view. So here we do have the DNA where it codes for the continuous molecule, and this is your primary transcript, and once it is spliced, you will be having the final processed mRNA. So the capping and tailing will happen hereafter, and what actually is important is here the splice junctions and the exon intron molecules. And over a period of time, evolution happens, means normally in human, the exonization of intron happens, and this normally happens just because of the presence of allo elements in that region. And that knocks the splice junction recognition sites. Okay, so I request you to go through this table again. And most of the time I said, like, just before this cell division itself, the duplicated molecule happens in its tetrad condition. So the very first stage of its division, it will be having its tetrad. So most of the time, like, we have discussed it, and we will continue this in the next revision class. We can stop here. Thank you. Any questions? If you want to discuss, you can post your questions in a message, comment. Shall we stop recording? Stop recording, please.
Ma'am, recording stopped.
Okay.